today's webinar. So we bring two expertise in neuroimaging infrastructure and also methodology, and highlighting how this capability can support our collaborative research, especially in Alzheimer's disease at Penn State. Okay, we have two speakers. The first speaker is Dr. Xiaoxiao, co-director of Human Imaging at Penn State Social Life Engineering Science Imaging Center in the Social Science Research Institute. Dr. Bai's work is focused on MRI, EEG, and fNIRS, right, with particular expertise in multimodal neuroimaging aggregation, processing pipelines, and also qualitatively cognitive analysis. So this we introduce the imaging capability and also the service available through the SLEIC and discuss how they can support the neuroscience and Alzheimer's research. Xiaoxiao, can you share your screen so you can take over whenever you are ready?
Okay, thank you, Dr. Hao, and I'm very happy has the opportunity to introduce our facility, particularly in the human image. So my colleague, Dr. Thomas Neuberger, he will present the high field animal part. So, okay. So why the neuroimaging is important to the Alzheimer disease? So because Alzheimer disease is a multiple system brain disorder. So the neuroimaging can provide the most widely used research and the diagnostic in vivo biomarker. So why we need to use the multimodal imaging approach, because no single neuroimaging modality can capture all the features of the Alzheimer disease. So here, this table lists the most popular neuroimaging module to study the Alzheimer disease. So include the structure MRI, diffusion MRI, functional MRI, and ASL, arterial spin labeling to measure the blood flow, vascular, and MRI spectroscopy, we can study the neurochemical imbalance. Okay, so we also have the EEG and fNIRS system can measure the neuron dynamic and the neurovascular coupling. The only major system, the pad, we don't have. So because the pad need inject the radiation contrast, so must you conduct in the hospital, in the clinical environment. So here I list the paper number if we check from the PubMed from the 2018 to the 2026. So you can see except the PET, we SLEIC, I mean the human neuroimaging facility, cover the most research purpose. So our SLEIC center located in the Chandler lab. So human part in the suite 6, 7, and fourth floor 120. The 6, 7 in the basement. So we have a two facility working for the human neuroimage. One is a 3 Tesla human MRI facility. So we have a 3T Siemens Plasma Fit MRI scanner. It is a high-performance human MRI system, can support the structure, function MRI, diffusion MRI, ASL, and MRI spectroscopy. We have a same as the hospital. We have an entire the coil. We can scan the whole body from the brain, knee, shoulder, neck, and abdomen. And we also have a research agreement with the Siemens. So we can free to get a lot of the work in progress sequence. In the development sequence, you can scan, you can use the most advantage technique. And we also installed ADNI4 neuroimage protocol in our system. Even we never scan the Alzheimer patient, but we definitely have this capability. We also has a lot of MRI compatible device. You can measure the cognitive function. You can measure the eye tracker, eye movement. And we also can measure the physiological signal like the pulse, ECG, like the motion and impedance. We can extend to the 16 module. And we also has like the response device. We have a simultaneous transcranial electrical stimulation for the brain. So we can try to study the increased the simultaneous for the different brain region or decrease the function connectivity for the different brain region for the cognition task. We have a second facility is a human electrophysiology facility. We have a high-density EEG with 256 channel. And we also have a GPS system can capture the real electrode location. So this function can make the EEG source image to project activity on the cortical brain. And we also can combine with our function MI, structure MI, spectroscopy, diffusion tensor image. We can run the multiple model imaging. We also has a high dense fNIRS system. So fNIRS system can measure the blood flow on the cortical surface. We have a 32 source and a 32 detector. So this system also supports the hyperscan. It means that we can scan the two people together. For example, we can scan the interactive task. We can, for example, like the mother and the child, or two people has like the emotion and play the card like this very complicated task. And we also has a neuron feedback software. We can measure the neural feedback. It means the online neural feedback and change the cognitive task, can test the imagination, the research field. With this high density EEG and high dense fNIRS, we also have the eye tracker and the transcranial electrical stimulation can simultaneous with the high dense with the EEG system. So actually, of course, we also build our data analysis pipeline in the ICDS, Raw High Performance Cluster. So we already, for the different module, we already build the different software in there. So each one can study the different features of the brain. So most pre-processing step, we can just run the one command line to finish the whole pre-processing. And for the others, we also have a script. So that means that our SLEIC, we supply for the human research, we supply the research support from the sequence design, sequence parameter adjustment, and the pilot scan. And we also support to the consultation for the data analysis, for the pilot scan, the whole procedure. We also working on and testing the machine learning tools in the raw cluster. We already tested the scikit-learn and PyTorch, those one. We try to make the imaging modality to the machine learning field for the traditional pipeline. We ask the ICDS to install the large language model, but we never test. The local model is already in the raw collab. The only reason is why we want the local version, because the human data has some data management problem based on the IRB. So we want to keep all the data storage and the management under the university field. So now I will show you some like the study we already finished and for each module. So first is the resting state function connectivity. So you know the resting function connectivity can measure the spontaneous, the bold correlation across the brain network, and very sensitive for the early network, like the disruption, for example, like the default network. And also it's a very good method for the Alzheimer disease study. This scan is in the ADNI default protocol. So we have a 64 subject. We do football player, and we do the pre-scan, post-scan, season scan, and then we search, we calculation the connectivity to the pre-season and the post-season, and then we compare the post-season and the pre-season connectivity, and we also check the correlation with the football player position and the concussion history. And finally, we can find which region show the difference. So this figure show the left precuneus has a significant difference function connectivity for the different position football player, speedman versus the non-speedman. And you can see the most non-speedman, the function connectivity post-season is an increase, but for the speedman is a decrease. So that is very sensitive for the whole brain neural network. So here is a method for the diffusion tensor image. So it names as a conductometry. That is recently very popular the method. So we just measure the local diffusion change along white matter pathway, and then those methods are very sensitive to detect the subtle and the focus white matter change or alternation. So we also can combine the white matter change with the other factor. For example, like the gender, age, or you have the clinical test score. And this method now also used for the Alzheimer disease. I give you an example for the football player. So we have 77 subjects, and also it's a pre-scan, post-season scan, and because a lot of the heat happen in the season. And then we run the diffusion tensor image analysis. We get the FA map. And then we can run the group analysis. We can combine the longitudinal change. We also can compare the position, our concussion history, and then we can find which tractor, which fiber region show difference between this condition. So the right figure show significant the decrease in the speed football player versus the non-speed people. So we run the correction statistic measurement, and then we build the fiber here. So that means that we know the how change before season and after season. So here, this one is another method for the diffusion MRI. It's called ALPS. It's to measure the glymphatic system function. So idea is we do the different data analysis, and we put all the data in the template space. And after that, we get the glymphatics, the region here. And then we calculation the ALPS, prevascular index number. And then we just change. We just compare the glymphatic system, how change before the season and after season. So here the method show the same thing. So the different position football player, the glymphatics after season and before the season has a significant difference. You can see the ALPS index show significant difference. So that's the method also used for the Alzheimer disease study recently. So here is a study. We used our high-dense EEG and combine with the function MRI and also combine with the machine learning. That is for the language. So we have a 20 subject. Sorry, this is wrong. So we have a 36 subject and 18 subjects speak Chinese and 18 subjects speak English. And we give the auditory task. And then during the task, we get the fMRI and the EEG separately. So from here, you can see we have the ERP. We have a 3-time peak. And then we get the 3-time map. And then we can, from the EEG source localization, you can see different peak show the different activity and amplitude. And this imaging, we use the fMRI result, the region, as a weight region. So it's you can call the fMRI limited image. And then, so we input this data to the machine learning, to the super vector machine. And then we do the training. Finally, we find the sum region, the different color means the accuracy. So you can see from the English, the accuracy region, highest accuracy region in here. But for the Chinese, in here. So that is the machine learning method we can use to measure to predict which part of the network most important for some specific function. So this one is ASL study. So ASL perfusion MRI can non-invasive measurement the cerebral blood flow of the subject. So very sensitive to the vascular and metabolic alternation. So actually, the study is like this. We have a 24 subject and 12 normal subject, 12 concussed subject. And we run the two scan. And during the scan, we have a cooling. We give the brain cooling 30 minutes. So we want to compare after treatment, what we change for the cerebral blood flow of the brain. So from this image, you can see the normal and the control, the pre-cooling is totally different. But after, if we compare the difference pre-subtract the poster, the blood flow, the normal is pre-last the poster, but that one, the control is different. So that can measure the treatment, how affect the blood flow. It's ASL is this method, blood flow method, also is very popular for the Alzheimer disease. This one is we use the fNIRS system. And we do a study. It's a chronic pain patient. So we run the 10 minutes resting state fNIRS, and then we give some like the neural nerve block treatment. And then we run another 10 minutes resting state fNIRS. So here you can see our fNIRS sensitive map. That means the dose region we can measure, get the very high accuracy signal. And then we calculation the correlation between the two channels. And then we find after treatment, we get the dose region show difference. So that is we can predict our treatment will affect which region and why they can stop the patient feel the pain after treatment. So can very good study the metabolic change from the brain. So that is the final, the modulation. We use the MRI spectroscopy. So also it's a concussion study. So that is the MRI compatible brain cooling study. So during the MRI scan, and then we run the brain cooling system. We reduce the brain temperature. And then we use the MRIs to find water and NA, the peak frequency location. And that like this. And then we can calculate the difference between these two. So we run the multiple scan during the cooling about the 30 minutes. So you can see here, the show very clear how the system reduce the brain temperature. So we not only can get the water and NA, we also can get the others, the component. We also can get the GABA. Those things is a very, very popular use for the Alzheimer disease, and also can use the others. I mean the neurodegeneration disorder or some like the cognitive development task. Okay, so that is all our human imaging capability. And thank you for listening. So if you have a question.
Good. Thank you very much. Let's see if we don't see the question in the chat box. So for the audience, if you have any questions, feel free to leave your question in the chat box so Dr. Bai can answer the question. So maybe I ask a very quick question. You do one language, one is English, one is Chinese. So actually, they are in the different region of the brain. So that means the different language we are using kind of different functions in different region of that brain. Is that true?
Yes. So that's the started idea is we know the for the language. So we assume the language network for the different people should be I mean in the same brain network. So our show here. So this is the function MI and get the brain network during this task. And then we want to know because I mean the EEG can give the very high temporal resolution. So we can know that during the task how the brain the change, but the function MI cannot give this one. So we want to know for the language processing, so the different language, which region most important to the specific language.
I see.
So that's why if you from here, you can see that the two is for the English. That's why if we use the machine learning to training, that's the model, we find that that's one is a very, very accuracy. That's why it has a normal feature for the English speaker people. And here is a very, very high accuracy after training for the Chinese, I mean the speaker. So we predict that's the two region is the most important for the one language and others for the another language.
Oh, very cool. And since you already have many data, I was wondering if that's possible. You have public data so people can access, do some secondary data analysis.
Yes, actually, so you see, we already build, I mean, this pipeline. So if we can download the public data, of course, we can run those, I mean, the data analysis for the secondary data analysis.
Right. But you already collect lots of data, MR data for football players, everything. Is can put public available, people can.
No, because the each data belongs to the each P for the one PI.
I
So
see.
only the PI, the principal investigator for this one, agree public or agree share. So you can do the secondary. So that is the two different the type of data. One type of data is already put in the some like the public, the website, like the open
Okay.
FMI or something. So we can download, we can just use. But like this, like the football concussion, like language data, most scan data belongs to the PI. So we
Yeah.
need to get
Okay.
the agreement with the PI.
I see. I see. So I think here is one question. This maybe go back to the language area we are talking about. Is that Broadman areas?
Yeah, I think here is very close the broken area. Oh, so sorry, you mean that this atlas is a Broadman? No, it's not the Broadman. It's another, the region of interest in the template. So uh if I don't know if I have we can talk the detail. I can show the detail maybe after the meeting if he has a or she
Okay.
has a more question because
Sure.
this is not the Broadman Atlas. It's a special. I think it's a HCP Human Connector template.
Okay. Okay. Very good. Okay, I don't see more questions here, or if you have more questions for Dr. Bai, you can just feel free to leave your questions in the chat box so we can come back. And so without further delay, so that we can let Thomas to give another talk, so that then we can go back to Q&A again. Thanks very much, Dr. Bai.
You're welcome.
Okay, our second speaker is Dr. Thomas Neuberger, director of high field MR facility at Huck. And Dr. Neuberger has contributed a wide range of interdisciplinary research projects and also has authored over 70 peer-reviewed publications and approximately 150 conference presentation. He will discuss about the high field MRI capabilities and their potential impact on advanced neuroimaging and translational Alzheimer's research. Thomas, can you share your screen? Then you can. Okay, good. Can you also unmute yourself?
Okay, so I'm unmuted.
Great. Yeah, please go ahead.
Yeah, thanks for the introduction and for the invitation. So we heard now a lot from Xiaoxiao about human imaging. I'm here. I'm running the high field MRI facility, and I will show you what we can do on the on the animal area. I have the presentation slightly different. Because Xiaoxiao was talking about a lot of all the different methods, and I thought I'm going to show you a little bit what those methods are actually. Let's see how far we can get. So maybe first an introduction. So we are also at Penn State, and we are in the same building as Xiaoxiao is with the 3 Tesla, which is kind of nice because we have all the MRI systems on one floor in Chantley. The high field MRI facility is a core facility, is one of the core facilities of the Huck Institutes of the Life Sciences, but we are also part of the Social Life and Engineering Sciences Imaging Center, the SLEIC. And here are our 2 web pages where you can find much more information. The one on our side, on the high field side, is getting overhauled, I think, hopefully soon, and we will have some more information there. Here at Penn State, we do have actually not only 2 or 3, we actually have 4 MR systems that can do imaging. And what is kind of nice is we have a 3 Tesla here where we can do human, we could do animals there theoretically as well. We have a 7 Tesla, a 14 Tesla, and a 20 Tesla. So we span a whole range of different field strengths. And therefore we will have lots and lots of different applications. So we can go from The smallest that we imaged was probably a canola seed, which has nothing to do with neuroimaging, and the biggest are humans, right? So it's a whole range. The high field facility is the 7 and the 14 Tesla MRI system here in Chantley. The 20 Tesla system is in chemistry and right now is mainly used for MR spectroscopy, not really much imaging, but theoretically you can do it. So our first system here is the 7 Tesla magnet. It's turn on. It still says Varian on it, but in reality, it's actually a Bruker system. So I don't know how many of you are actually working with animals or are interested in animals, but so it's not Varian anymore. So Varian doesn't exist anymore. It's all Bruker. It's up to date. It's the newest system. It's a Bruker BioSpec 7030. We can put in here usually mice. We have birds in there. We can do rats, and they can be all in vivo, or we can also do ex vivo studies. We have home built and also commercially available resonators, and this system has 4 receiver channels and 1 receiver channel for X nuclei. What does X nuclei mean? We do not only need to do, we can also do other nuclei, not only protons. We can also image sodium or phosphorus or anything else that you might be interested in, and it's NMR sensitive. The 14 Tesla system looks like this here. It's a vertical system, so it's a little different setup. For neuroimaging, we can do all of the methods that are available. The only thing is, I guess, most of the neuroimaging community went away from the vertical systems because the animal has to be in a vertical holder. So it's not horizontal, it's vertical. So that's why it's usually not used a lot for fMRI, not at all used for fMRI. But this system here is only one in a few in America. There might be a handful of those for imaging in the US. And what you see in the right corner in the room here, it's a so-called cryoprobe, which I will show you some images from. That's actually the only one in the world right now that is working at 600 megahertz. So for our 14 Tesla system. Besides MRI, the high field MRI facility, we actually do have also a CT scanner here, and that's a micro CT scanner from Bruker. And that is nice because it can image bones, which means we do multimodal imaging. You can image the soft tissue with MRI and then the bones with the CT scanner, and you can fuse them and you can get nice overlays, which can be helpful in some studies. So besides our main instruments, we also have, of course, areas where we prepare the animals for scanning. We have all the animals need to be monitored during scanning. We have all of this available in the facility, so we can measure their breathing rate, theoretically their blood oxygenation level, and their heart rate, and of course the temperature. And also what's nice in our case here is we can build our detectors for whatever we want to image here ourselves. So we have the knowledge, and we did build quite a few of those detectors ourselves. And also the animal beds. We have a 3D printer here, so we can print anything that we need to hold animals, for example. And of course, we have enough computers and all of these things. This here are some of the resonators that we built over the years. You can see they are not really pretty, but it's in MRI, it's very essential that your resonator is about the sample that you want to image. So for example, in this solenoid coil, you wouldn't image a mouse, of course, but here for this surface coil, this would fit perfectly onto the head of a mouse, and that's what we used it for, right? And then these are bigger resonators than for the 7 Tesla system. I do have a special one here. So this is this cryoprobe that I mentioned before. Maybe a word for cryoprobe. What does this mean? So in MRI, the signal to noise ratio is very important. And to increase the signal to noise ratio, a lot of effort is taken. And this is one approach. So what we are doing, we are not increasing the signal with this thing, we are reducing the noise. So by making this cryoprobe cold down to 20 Kelvin, we have no noise from the electronics. We only get noise from our sample, and therefore our SNR is going up at least by a factor of 3. And this gain in SNR then can be used to increase the SNR and the resolution, sorry. Yes. So why do we do MRI? MRI is safe. It's better than other modalities like CT scanning. You have ionizing radiation. And yeah, MRI is in the hundreds of megahertz. So that's right where your radio station is. And it's still far away from the microwave oven. So MRI is very safe. And we can get very nice contrast. And just by changing the imaging parameters, we can get different contrast inside the tissue or inside your animal in this case. You can see the bright area here, it's the CSF, and the eyes, it's bright here and it's kind of darker here. Okay, in this newer software that we have, we already have some AI built into the data reconstruction. This is exactly the same data set. On the left-hand side is the original one, and on the right-hand side is the same data set with some denoising applied. This is on a mouse brain, and this is on a turkey brain. So you can see it's increasing, or it's improving the image. Maybe a little too much. I'm not sure if all the details are still visible, but that can be still adjusted. So that's already here. And then also there are some of the newer techniques, like that's called compressed sensing. This is a technique that's not acquiring the full data set. It's kind of leaving out some lines in K-space, where the experts know what K-space is. It's using some wavelets to reconstruct the data. A regular scan would be 22 hours, and by using this technique, we can actually go down to 4.5 hours and get still a decent image. If you look in detail, you see it's a little bit deteriorating, but not that much. It's still fine. Okay, so this was kind of an overview what we have here. And I mean, if you have any questions about this, please let me know. And then again, this is kind of the same things that Xiaoxiao was kind of talking about. I just wanted to show you some examples how this looks like in mice. This one is the T1 and the T2 weighted. That's what I just pointed out already. Other structural imaging is, this here is the stuff that we've done a long time ago. But this here is a spinal cord injury in a rat brain. And you can really in vivo, this is the aorta, by the way, you can in vivo nicely see how the damage, where the damage is. And then you could theoretically, again, it's MRI over time, you could see if this could be repaired with some method. So, and we can also look at some diseases. This is an experimental autoimmune encephalomyelitis. And you can nicely see the bugs that are in here, the lesions, right? This one is ex vivo. And this is where we are with our cryoprobe right now. If you compare it to histology, it looks already pretty similar. And then maybe, let me see if I can, yeah, maybe if I can get this done. So this here is our scanner, and here you can actually see this data set. And what's interesting here is in histology, you have to decide which direction you want to slice your brain, right? Your mouse brain. In our case, I can decide in which direction I want to look at it, and I can look at it again in another direction, and I can do whatever I want. It's a digital data set. This particular data set, I think, has an isotropic resolution of 8 micrometer. So it's getting really close to histology, and a lot of things can be seen in here without any special imaging. This is just structural imaging, right? So we can play around and go through it. And I mean, for the ones of you who have done some histology, I hope they can appreciate this here. Okay, let's go back to the PowerPoint. This is structural imaging. I mean, I do have some papers here that I don't want to go into details, probably don't have enough time, that are kind of using structural imaging in in Alzheimer's disease, but you can look this up maybe later yourself. What's used a lot in as well is contrast-supported magnetic resonance imaging. That's where they use gadolinium, for example, to increase or to speed up the imaging time, or to increase some special features inside the brain. Okay, so that's structural imaging. Flow MRI for Alzheimer's disease, I'm not so sure if flow, like the larger blood vessels, are affected, but we can do image, for example, the blood vessels inside a mouse brain. So in this case here, they were actually studying stroke, and they were occluding the ICA with ET1. ET1 was injected, and they were not really seeing if it worked or not. By doing this angiogram, you inject the ET1, and you did do a scan before, and you see nicely after the injection, the whole blood vessel here is gone. And you can nicely see this in a living animal, and then you can also see that after 14 days, the blood vessel kind of is back again, and it's, yeah. So that's a nice feature. Okay, talking about functional MRI. I don't want to talk much about functional MRI. Xiaoxiao did talk a lot about it, and but what we are seeing here is, I don't know if maybe I don't even have to go into the detail, but it's kind of, you're looking at the bold effect, right? If you start finger tapping, your motor cortex starts to work, your motor cortex says, I need more blood, and then we have more blood going into this area, and then therefore we kind of see an increase in the area. And it also has to do, oxyhemoglobin is diamagnetic, and therefore it's giving us more signal than deoxyhemoglobin. And this is kind of what we are looking at in fMRI. This is the pulse sequence for fMRI, and yeah, we have here, it's not a sequence that is straightforward. There's a lot of reconstruction necessary, and a lot of pre-processing even to see this. But here on campus, on our 7 Tesla system, we do have Dr. Danying Zhang, working on it, and he's a real expert in this, and I'm sure he's willing to collaborate with a lot of people if they want to do fMRI. And okay, I don't want to go too much into fMRI. Next thing, here again, here are some papers. The next thing is perfusion measurement. That is also used a lot in Alzheimer's. We haven't done this on animals yet here, but we can do this without any problems, I would say. And the way what it's done is you're kind of acquiring 2 different images, and one has you're saturating the blood before it's entering the brain. And then you're doing 2 images, one with saturated blood and one with unsaturated blood. And then you can calculate what the perfusion of the brain actually is. Here again, some papers for you to look at later. And then next thing that's interesting is, of course, the diffusion tensor imaging. And in this case, I mean, we have to go a little bit more into detail. So if you know, diffusion is happening everywhere. If you have, just a glass of water, your mean square displacement of the molecules is written here. It has the diffusion constant and the time. The longer the wait, the further the molecules are away from each other. We can use this in MRI, this technique. And in MRI, our signal is then weighted with this function here. This function here contains an apparent diffusion coefficient and also a B value. The B value is this guy here. This guy consists of, this is a constant, our gradient strength that we are using. So it's I think I shouldn't go into these. So it's specific on how you switch your gradients inside. So the higher the B value, I mean, that's the important thing. The higher the B value, the less signal you will have if you run this. And if you run this here with switching those gradients in different directions, you get you can actually follow the diffusion of the water molecules inside the brain. If you think about the isotropic diffusion, what I just said, it's going everywhere. But in the brain, you have white and you have gray matter. Gray matter is more dense. Something like this here is more dense. And a water molecule is kind of not diffusing through your fiber bundle, it's kind of diffusing along the fiber bundle. And when it's diffusing along the fiber bundle, it's more moving in this direction. And we can detect this by actually calculating this complete diffusion tensor. And if we have this tensor, we can then, let's skip this, we can calculate different things. One thing is the fractional anisotropy, that's written here. So as I mentioned, the diffusion is going in one direction. If it's only going in one direction, the lambdas here are the eigenvalues. If it's going only in one direction, then we have only one lambda 1 would be 1 and the others would be 0. So that would be ideal. So the FA map can be between 1 and 0. If you have directed diffusion, it's 1. If you have isotropic diffusion, it's 0. And again, we can do this in the mouse brain, and this is how it looks like. So this is kind of the FA map. I just try to explain to you here. So if the brighter the image, the more directed diffusion we have, right? We see in the corpus callosum here, it's kind of white because the FA is kind of high, and then in the gray matter, it's kind of dark because the FA is lower. And this can be used then in different mouse models, right? If you have different disease, it's shown that in Alzheimer disease that the FA actually changes in certain regions. And with this information here, then you can actually do some fiber tracking. And I do have, I think, if I go to the screen here, so this is the FA map again of the mouse. Go through it. It's a relative high resolution DTI data set. I think it's only 35 microns. And then if I turn on tractography here, and it looks something like this here, right? So you get this tractograms from the whole mouse brain. And then again, you can compare this here to either diseased or to healthy animals. Need some information. Okay, let's move on. The next thing that we can do is MR spectroscopy. So MR spectroscopy, you might have done NMR in chemistry before, where they do only spectroscopy, but in this case, they are getting a spectrum from the whole volume. We can do spectroscopy at only a certain area in the brain. So we can decide, we use this voxel here, and I want to see what is the metabolic profile in this area here. And then we get something like this here. That was on the 7 Tesla. This is what we should get on the 14 Tesla. So you can nicely see the NAA, creatine, choline, taurine, all of those guys are all here. GABA is here as well. It's a little hard to image GABA, but there's a special method that is only made to detect GABA. This often, so the GABA guy here, right? This here is, you can do this then again on healthy animals. You can do it in different areas of the brain. I mean, it's known that NAA is not constant over the whole brain. It's different in different areas of the brain. And then, for example, choline is mostly in cell walls. So if you have cancer, if you're looking for cancer, you will have an increased choline peak wherever the cancer is. So it's a powerful method. Here is a list of what can be found in the brain with MRI without problems. It's relatively long, and yeah, you can look at this after the talk. Here again, just some papers. And the last thing that I want to show you here, it's called chemical exchange saturation transfer. So this is relatively new, and this here is to detect things that we usually cannot see in spectroscopy. So it's looking at the exchange of protons. the fast exchange. So we are actually exciting the protons at frequencies that 4.7 is our water frequency, but we are exciting somewhere here. So the spins are getting excited, and then they are kind of traveling over and get attached to the water. And so the water and whatever is here are exchanging protons. If they exchange protons, the signal from the water will get a little bit lower. And this is what's shown in the C spectrum here. So we can actually detect this guy here much better if you look at the so-called C spectrum. And we have water, of course, it's going to 0 then. And this is a way to detect, for example, amide protons that you couldn't see in regular spectroscopy. So it's a very powerful tool and is used quite a bit. I mean, here are some papers, and it's a relatively new tool as well.
Thomas, maybe we
Yeah.
can finish in 1 or 2 minutes. We can leave some
No,
time
I'm
for
finished.
questions.
I'm finished. So there's a whole plethora of techniques that we have here. The last thing I want to show you is the CT scan here. And this is what I said before. We get MR soft tissue, and we get the bones, and then you can fuse it together. And so that's all that I want to tell you today. I mean, I know it was a little much, but I think I hope it helped a little bit. And I don't know if you have any questions.
Okay, great. Thank you very much. It's I don't see the question in the box, but feel free to write any questions for either Thomas or Xiaoxiao, and we can discuss. And Thomas, I have one question: is when you because you talk about diffusion over there, right? So you said okay, you compute the eigenvalue, then see how the eigenvalue. I was wondering, this diffusion could be different over the space, right? So
Yeah.
if you look at the eigenvalue, could be a function of the space as well.
So we are calculating diffusion tensors in every single voxel, right?
Okay.
So every single voxel has a diffusion tensor, and in there we are then calculating the eigenvectors and the eigenvalues and have this for each single voxel. And that this is a way then actually, if you look at the eigenvectors, you can actually trace the connectivity of the white matter, for example, right?
I see.
From one voxel to the next voxel and see how is the change in my biggest eigenvector, and then you can just follow it. And this is how you do your fiber tracking. And then
I see.
you would see if something is disrupted, for example, in one case, and that's the way how
Okay.
it
Okay.
is.
So in that case, is from mathematical modeling point of view, so we do have a diffusion equation, right? So
Yeah.
is that possible? You can just use the diffusion equation to model everything to see how the diffuse. This is essentially okay. Okay. So for you mentioned structure MRI. So structure MRI can also measure amyloid beta.
Yeah. And it's
So
usually
that
they use usually then even some contrast agent which will enhance this contrast then. That's all what we want to have in MR is this contrast. So you have some gadolinium, and there are other studies who actually use iron particles to get a dark contrast, right?
Okay. But people normally use that based on PET scan data, right? So then what's the difference? So you can use a PET scan to measure what's the amyloid and also use the SMRI.
Yeah. So we did talk about a PET system here as well at some point to get one. There is one available that would fit in our 7 Tesla system, but we didn't get enough people here who were interested in it. But in the disadvantage of the PET system is still the resolution, right? And of course, it's a little hard in our area to get because you need some radioactive material, right? And, but it's possible. Disadvantage is just low resolution. So therefore, the combination of PET and MR is ideal, right? Because you get the high resolution of the MR, but you have only the low, but the sensitivity of the PET, right?
I see. Very good. Okay. I don't see any questions in the chat box. I think that's probably is let's conclude our today's seminar, and thank you very much for our two wonderful speakers, and also thank everyone joining us. So down the road, we will have other, if you have any other suggestions for Alzheimer's disease or any suggestion for webinar, which theme or which topic we need to focus on, so please either send me an email or send an email to Camelia or Brittany. Okay, thank you everyone, and we will see you next time. Okay.
Thanks.
Bye-bye.
Bye.